First of all, thank you so much for inviting me to such an exciting symposium. It's been a lot of fun. I started my lab last year at the UW Pharmacology, and the focus of my lab is mitochondria, calcium signaling, and metabolism. So before I begin showing you data, I would like to give you a brief overview of mitochondria in general and calcium signaling and how they relate to each other. I'm sure you know that mitochondria are very important. They're powerhouses of the cell. They generate your ATP. And they're also very important for cellular metabolism. Approximately 1/3 of all human metabolic reactions can take place in the mitochondria, and 1/5 of them exclusively take place only in the mitochondria. However, it will be doing injustice to these mighty organelles to say that they're only important for metabolism. They have many other cellular functions. They are important for cellular immunity. They regulate cell death. They are important for peroxisome biogenesis, and they are they play central roles in protein homeostasis and calcium homeostasis. Of course, one thing that we always have to remember is that mitochondria do not perform all these functions in isolation. They have to communicate with the rest of the cell, and they have to be very versatile, and they have to respond to cellular needs. And a prerequisite for this versatility is effective communication between mitochondria and the rest of the cell. So how is this communication between the mitochondria and the rest of the cell mediated? There are many ways of doing this communication, and I'm going to simplify ways of doing this communication in this slide. Mitochondrial functions can be regulated by post-translational modification of mitochondrial proteins, or mitochondrial gene expression can be regulated to alter its function, or small molecules can be used to alter mitochondrial functions. In return, mitochondria has to communicate its own status to the rest of the cell, and it does so by using its decreased membrane potential as a signal, and sometimes metabolized use in the mitochondria can also be used to signal its own status. We think that calcium is a very important mediator of this regulation between the cell and the mitochondria. In fact, there is a very intricate relationship between mitochondria and calcium that has been recognized for a long time. So the first part of this intricate relationship is regulation of cellular calcium signaling by mitochondria. And I think this point is best illustrated by these videos that I would like to show you. In these videos, you will be looking at a section of a xenopus oocyte, which has a calcium indicator dye in it. At the beginning of the experiment, IP3 is injected, which initiates a release of calcium from ER stores, and it starts a calcium wave in the oocyte. Let me see. And then this is what happens. This is calcium signaling and calcium waves in the oocyte. Later in the experiment, mitochondria are energized, which leads to calcium uptake by the mitochondria. I think you can appreciate even by looking by eye that the calcium signaling events and calcium waves are dramatically different in the absence and in the presence of mitochondrial calcium uptake. And this data is quantified here. Mitochondrial calcium uptake leads to synchronization of cellular calcium waves, and it also increases their amplitude, which can have dramatic effects in downstream calcium signaling. So this is what happens to calcium in cellular calcium when mitochondria are involved. What about the mitochondria side of things? So if you actually look in detail, we realize that mitochondria are calcium-regulated organelles. First of all, there are 13 calcium-binding EF hand-containing proteins in the mitochondria, and 2 of these proteins regulate mitochondrial motility through their EF hands. The rest of the EF hand-containing proteins are all metabolite carriers or ion carriers, and the rest of them, together with calcium-regulated dehydrogenases of the TCA cycle, regulate mitochondrial metabolism. In addition, calcium overload in the mitochondria is a stress signal, and it can lead to loss of mitochondrial integrity, which can eventually lead to cell death. So now the question becomes, how does calcium enter the mitochondria? Given the importance of this mitochondrial calcium uptake for regulation of metabolism, calcium signaling, and cell death in general, there was a lot of interest in trying to understand this mitochondrial calcium uptake process. And this phenomena has been studied for a long time using biophysical methods. And many years of research using most biophysical and in vitro methods has established that calcium enters the mitochondria via a uniporter that resides in the inner membrane of the mitochondria. This is called the mitochondrial calcium uniporter. And like I said, it resides in the inner membrane. And this uniporter is highly selective for calcium. It doesn't let any other ion go in. And mitochondrial calcium uptake is dependent on mitochondrial membrane potential, intimately linking mitochondrial health to mitochondrial calcium uptake. In addition, again, years of research has suggested that mitochondrial calcium uptake is important for many cellular processes as well as many disease states. However, a causal role for mitochondrial uptake in involvement of these processes was not possible to test because the molecular identity of this uniporter remained a mystery in the field for a long time. Even though people tried to identify the uniporter for more than 60 years, they couldn't find it. In 2010, my postdoctoral laboratory identified the first protein that was ever involved in mitochondrial calcium uptake, and they did so by using clues from evolutionary biology, from evolution. Years of research had established that yeast Saccharomyces cerevisiae does not have any mitochondrial calcium uptake activity in its mitochondria. And this is a typical calcium clearance assay by the mitochondria, which I would like to introduce. In these experiments, mitochondria are isolated, and they are incubated in the presence of a calcium indicator dye, and the calcium is injected. Because there is no calcium uptake by the mitochondria, we don't see a decrease in signal intensity. And this is what happens in yeast. And again, we knew for a long time that other unicellular species, in this case Leishmania, had mitochondrial calcium uptake activity. This is clearance of calcium by mitochondria. So a postdoctoral research in my previous lab decided to compare the genomes of cerevisiae to that of Leishmania or other kinetoplastids. And they found that when compared, there is only a small number of genes that lack homologs in yeast. And by doing a targeted RNAi screen, and at that point, it wasn't even a screen, it was a small RNAi experiment, they found a protein that altered mitochondrial calcium uptake when knocked down. And this protein was called MICU1 for mitochondrial calcium uptake 1. However, and this MICU1 has 2 EF hand domains. However, it was immediately clear that MICU1 was not the uniporter that they were looking for because MICU1 is not a transmembrane protein. It resides in the intermembrane space of mitochondria, so it couldn't be potentially the uniporter they were looking for. But using MICU1 as a molecular handle, they could identify the uniporter itself. A graduate student looked at what has co-evolved with MICU1 and also what was co-expressed in MICU1 in different databases. And this is the co-evolution data, and this is co-expression data. On top of both of these data sets, there was one gene that was called CCDC109A. This protein had 2 transmembrane domains. And it also had certain residues that were typically found in calcium transporters, very acidic region, very acidic domains here. And we also showed that it resides in the inner mitochondrial membrane of the mitochondria. So when I joined the lab, we decided to check whether these 2 proteins interact. And we saw that MICU1 and MCU, when we pulled down MICU1, we saw that, when we pulled down MCU, we saw that MICU1 interacted with MCU, and suggesting immediately that the uniporter is probably not a single protein, but is a bigger protein complex. And again, by using computational methods, we found another protein, a homolog of MICU1, MICU2, And similar to MICU1, MICU2 also had EF hand domain containing EF hand domains, which are calcium binding. And we show that similar to MICU1, MICU2 also immunoprecipitates with MCU. So at that point, I'm going to summarize a lot of data from different labs, including ours, and show you our working model, our model for MICU1 and MICU2 function. So as I told you, both MICU1 and MICU2 reside in the intermembrane space of mitochondria, and it turns out that they are gatekeepers for mitochondrial calcium uptake. And this is a very important function because, like I told you before, mitochondrial calcium overload can be detrimental for the cell. So it's very important to make sure that there is no calcium overload in the mitochondria. And we think that in the presence of low cellular calcium levels, MICU1 and MICU2 associate with the complex, and they prevent mitochondrial calcium overload. When there is an increase in the cytosolic calcium levels, they bind to calcium through their EF hands, and then this leads to dissociation of MICU1 and MICU2 from the uniporter, and then calcium is now free to go into the mitochondria. And we showed that this calcium uptake threshold for the mitochondria is around 1 micromolar, which is the calcium affinities for these EF hand domains. So, of course, these were very interesting findings, and they were very exciting for the field, but we still did not know what the calcium conducting pore of this uniporter complex was. Of course, MCU was the most likely candidate. It is a transmembrane protein. It has 2 transmembrane domains, and it had certain residues that were important for calcium uptake. So we wanted to see whether MCU is actually the calcium pore. So we decided to reconstitute uniporter activity in yeast. I told you that yeast doesn't have any mitochondrial calcium uptake activity. So we expressed MCU alone in yeast, and we saw that it goes to mitochondria properly, and it forms a big molecular weight complex, which is around 240 kilodaltons in a BN page. However, when we isolated these yeast mitochondria and we did a calcium clearance assay, we showed that there was no calcium uptake in these yeast mitochondria that express MCU. So this immediately showed that MCU by itself is not sufficient to form the uniporter, a functional uniporter. And it suggested that, so at this point, like all the evidence suggested that MCU must be the pore, but we couldn't reconstitute mitochondrial calcium activity by expressing MCU. And we wondered whether there was a missing component in this uniporter complex. So at that point, we were actually, we had exhausted all the computational methods for finding out other components of this complex. So we had to move to doing biochemical characterization. So to do biochemical characterization, I expressed MCU flag in 293 T cells, and I could purify a protein complex that has the same molecular weight as the endogenous complex. And we did a mass spectrometry characterization of this purified complex, and we found that there were only 5 proteins that were highly enriched in this mass spectrometry experiment, and they were, of course, MCU and its paralog, MCUB. We also found MICU1, the regulator, and MICU2, the other, its paralog MICU2. And we found another protein that wasn't characterized before. It was known as C22 or 32. And we named this protein EMRE for essential MCU regulator. So since EMRE was the only new protein that we found, we decided to characterize it further. And first, we made sure that this is a bona fide interacting protein by showing that it specifically interacts with MCU, but not a control protein, similar to other complex members. And again, similar to other complex members, EMRE is ubiquitously expressed in all tissues that we checked. So EMRE is a very small protein, approximately 10 kilodalton, and it has a predicted transmembrane domain. We wanted to see whether it was a real transmembrane domain, So we did carbonate extraction experiments and tried to pull it off the membrane by using high pH buffers, but it stayed in the membrane similar to MCU, and this is a controlled soluble protein. And EMRE has a very interesting C-terminal domain that is conserved, which we call this C-terminal acidic domain. And in experiments that were not published yet, we showed that the C-terminal domain faces the intermembrane space of mitochondria, and we think that this region is important for interacting with other members of the complex. So, and in the interest of time, I'm going to skip over a lot of data, but I'm going to summarize and tell you that we showed that EMRE has two functions in this protein complex. One is that it is absolutely essential for mitochondrial calcium uptake activity. If you look at MCU EMRE knockout cells, we see that there is absolutely no calcium clearance, whereas wild-type cells take up calcium. And in other set of experiments, we realized that in EMRE knockout cells, the MCU protein complex has a much reduced molecular weight, more so than you would expect by losing a 10 kilodalton protein. And this suggested that maybe EMRE is mediating the interaction between MCU and MICUs. And to check that, we purified MCU from EMRE knockout cells, and we saw that the other components of the complex do not associate with MCU in the absence of EMRE. So of course, at this point, we wanted to see whether EMRE was the missing component that we were looking for in our experiments. So we decided to go back to our yeast reconstitution experiments. And this is the data that I showed you before. When we express MCU alone, we don't see any mitochondrial calcium uptake activity. But when we express EMRE together with MCU in yeast, first we see that the size of the protein complex increases a little bit. And second, sure enough, when we did a calcium clearance assay, we saw that we could reliably reconstitute mitochondrial calcium uptake activity. So these experiments show that both MCU and EMRE are necessary for calcium uniporter activity and in these assays. So, but still, there is the question of what does protein do? What does EMRE do? I have to say, this lipoprotein was actually very confusing for us for a long time, because it turned out that not all MCUs require EMRE for calcium conductance. In fact, when we looked at Trio Life and annotated the species based on presence of MCU, MICU, or EMRE homologs, we saw that EMRE is a metazoan-specific protein. And for example, the slime mold dictyostelium discodium has an MCU homolog. It doesn't have an MCU EMRE homolog. But when we express DdMCU in yeast, we can reconstitute functional uniporter activity only by itself. It doesn't require EMRE at all to function. So these were very confusing, but we thought maybe we can use this confusing results to our advantage and try to understand what is different between these two proteins, between dictyostelium MCU and human MCU. And if we understand those differences, maybe we will understand how EMRE is functioning in those cells. So we wanted to know why human MCU requires EMRE, what makes DdMCU EMRE independent. So we thought maybe we can just start by comparing these two species in terms of their secondary structure. And this is a secondary structure prediction of both DdMCU and human MCU. And I think you can appreciate that these two proteins are quite similar, especially at their C termini, but their N termini are quite different. So we thought there must be something in the dictyostelium N terminus that make it EMRE independent. And then we thought maybe we can take this N-terminus domain of dicty, give it to human MCU, and make it EMRE independent. So to do these kinds of experiments, we needed an expression platform and experimental platform. And we decided to work with MCU knockout cells and EMRE knockout cells. So we made these chimeric proteins between these two proteins. And then we expressed them in MCU knockout cells to see whether they will be functional proteins, and in EMRE knockout cells to see whether they will function independent of EMRE. And this is the first two chimeras we made. And our prediction was that chimera-1 will function independent of EMRE because it has this region from DdMCU. But we actually got the exact opposite result. To our surprise, chimera-1 still needed EMRE to function, whereas chimera-2 functioned independent of EMRE. So these were very exciting, and we thought maybe we can keep doing these kinds of chimeric experiments and understand why human MCU needs EMRE. And we made more of these chimeras, but I'm not going to show you the data from these, and I'm going to refer you to Melissa's poster, because she has a very good story, and now I think we understand why human MCU needs EMRE. So I encourage you to go check out her poster to see the rest of the story. So at this point, I'm going to go back to this slide that I showed you before. I mentioned that years of research has suggested that MCU or the uniporter was involved in many of these processes or diseases. But nobody could really test those hypotheses because the molecular identity of the uniporter was not known for a long time. Now that we know many of the components of this protein machinery, the field has been perturbing mitochondrial calcium uptake by changing the expression of these proteins. And not surprisingly, maybe in an expected way, we learned that many of these proteins are essential for many different functions, and they are perturbed in different diseases. And of course, I'm not going to go through this data or this list, but this is just to show you that this protein complex ended up being very important for many cellular functions. And it's also highly regulated in different contexts. And our lab is interested in understanding what happens upstream of this complex and why perturbing the expression of these genes lead to these phenotypes that are observed. So with that, I would like to thank my new lab, especially Melissa, for taking over the Chimera project and doing a very good job in finishing it. We're hoping to publish it soon. And I would like to thank you for your attention. I'm happy to take any questions.
We have time for maybe one question.
It was very exciting talk. My question is very rudimentary. Maybe I have missed. Are they all nuclear encoded genes? None of them is mitochondrial encoded.
They are all nuclear encoded. All of the uniporter components are nuclear encoded.
So is that one way that how the cell try to manipulate mitochondrial activity or function, kind of try to tame it in a way?
So their expression seem to be highly regulated in different contexts, as well as the protein stability of this component. So yes, I think there is the cell is trying to regulate mitochondrial calcium activity by regulating the expression of these genes.
All right, thanks.
Great. Thank you so much. All right.